Hi everyone, welcome back to our lecture on molecular cloning methods. In the last part of the lecture, we talked about plasmids as vectors for molecular cloning. In this part, we will discuss phage and phage-derived DNA vectors. A very efficient way of getting DNA into cells is to use bacteriophage, the viruses that infect bacteria. Phage viruses are highly evolved to introduce their own DNA into bacteria. We can regard them as efficient DNA injection machinery. The idea is that you can replace some of the viral DNA with the DNA that you want to propagate. One example here is lambda-phage. It is a natural vector that propagates DNA in bacterial cells. In lambda-phage, the genome can exist in a linear or circular form. When this bacteriophage infects the E. coli cell, it injects linear DNA that then circularizes in the cell, and this either can get integrated into the chromosome of bacteria, or it can start to replicate to produce linear pieces of DNA that can be packaged into phage particles. The DNA is replicated by a specialized replication process called rolling circle replication, also called sigma replication since the replicating DNA resembles the Greek letter sigma. This generates a linear piece of DNA that has many copies of the phage genome, including copies of a specific sequence called a cos site. The phage proteins recognize cos sites, cleaving them to produce individual linear phage genomes, which they then package into phage particles. In order to use these bacteriophage as cloning vectors, you can take the linear phage genomes and engineer them so that a piece in the middle is cut out and replaced with an inserted piece of DNA that you want to clone. This works because the DNA that is cut out of the phage genome and replaced is not necessary for the virus to replicate, and the phage proteins that package pieces of DNA into phage particles recognize stretches of DNA that are flanked by these cos sites as long as the two sites are the appropriate length apart. So a phage particle can only accommodate DNA of a certain size, and this allows you to clone fragments of DNA that are about 10 to 20,000 base pairs in length. Phage can be vectors for much larger pieces of DNA than what you could typically clone using plasmid vectors. When the phage vectors infect bacteria, they can replicate virally and produce many phage particles, killing the bacteria in the process. The cloned DNA is copied along with the viral genome. Shortly after the infection, the mixture of bacteria and phage-carrying cloned DNA are spread on an agar plate. The bacteria grow, but where a cell had been infected with a phage, you will begin to see an area of dead cells from which the virus is spreading. These are called plaques. Each plaque is rich in phage particles carrying identical copies of a cloned piece of DNA. Cosmids are another kind of DNA vector that have some properties of phage and some properties of plasmids. A cosmid is essentially a plasmid into which the cos site of a lambda phage has been cloned. So the result is a plasmid with a cos site. We can cut this cosmid with a restriction enzyme and also cut large pieces of DNA, for example from an organism's genome, by partial digestion with a restriction enzyme that gives sticky ends compatible with the cut cosmid vector. If you ligate this mixture together, you get long pieces of DNA with cos sites randomly interspersed between stretches of genomic DNA that you want to clone. You can then add phage components. The phage proteins will recognize the cos sites and chop up the DNA into pieces. If the pieces are the appropriate size, they will be packaged into phage particles. Then, these phage particles can infect cells and introduce DNA, which then circularizes inside the cell, and because these cosmids do not encode the genes for phage proteins, they do not produce any phage particles. Instead, the cosmids with cloned DNA inserts replicate inside the cells as circular DNA from an origin of replication. No viruses replicate, so you are not looking for plaques, you are looking for colonies that grow on plates with an antibiotic that can select for the marker on your cosmid, in the case of this example, tetracycline. We have other kinds of phage and phage-derived vectors as well. Your textbook describes some of these, but I won't go into very much detail here. There are M13 phage vectors. These are a filamentous phage, and one key feature is that they maintain their genomes as single-stranded DNA. So if for whatever reason you want to isolate cloned DNA as single-stranded DNA, these M13 phage vectors can be used for that. Like Cosmids, there are other plasmid-type vectors with phage-like features. Plasmid vectors derived from M13 phage are called phagemids. They have two origins of replication. One plasmid origin of replication allows it to replicate as circular double-stranded DNA as a normal plasmid would, and another origin of replication, an F1 origin coming from the filamentous phage, allows the DNA to be replicated as single-stranded DNA, which can then be packaged into M13 phage particles. I won't go into much more detail here, just to introduce that there are other types of phage-derived vectors. We introduced plasmids and phage as different types of DNA vectors, and these have different applications. Plasmids are normally used for cloning relatively small pieces of DNA compared to other vectors. This is usually 10 kb or less than 10,000 bases. This would be, for example, to clone a single gene, or small fragments of DNA in order to manipulate single genes. Phage and Cosmid DNA vectors are useful for cloning larger pieces of DNA, 10 to 40 kilobases in size. These are usually for cloning genomic libraries. You can make a genomic library from the genome of an organism by chopping up the DNA and cloning it into a phage or Cosmid. You will get different clones that each have a specific piece of the DNA from the genome of that organism. Collectively, in that library, you can clone an entire genome of that organism. The library will consist of different clones which each carry a part of the genome. Starting with copies of genomic DNA from many cells, when you produce a genomic library, you will end up with overlapping fragments in different clones that span the sequence of the entire genome. You could amplify and isolate individual clones carrying the DNA of interest. That could be useful if you are studying a specific gene or set of genes. There are other types of DNA vectors for much larger fragments of DNA and for other applications, but I won't get into those. Through cloning, you can generate different types of libraries. We can build a genomic library, as we briefly discussed, which is derived from the total genomic DNA fragmented into pieces. Another kind of DNA library we will talk about later are cDNA libraries. cDNA is complementary DNA that is converted from mRNA sequences through a process we talked about briefly called reverse transcription. cDNA libraries are useful for studying only the transcribed regions of genes that are expressed. We will save discussion of these for the next lecture, but we'll first focus on genomic libraries. A genomic library is a collection of clones that, together, cover the entire sequence of an organism's genome, with many clones overlapping in regions of the sequence. Genomic libraries are commonly used for sequencing applications, playing an important role in deciphering the genome sequences of different organisms. As we discussed, phage and Cosmid vectors are very useful for producing genomic libraries. If you are going to prepare a genomic library to clone into a phage or Cosmid, what you would do is you would isolate genomic DNA of the organism, and then you would cut it into appropriately sized fragments depending on the vector. So if we're talking about lambda phage or Cosmids, then this will be tens of thousands of base pairs. Then you would ligate the cut fragments into your vector. So how do we get fragments of the appropriate size? We can use restriction enzymes to cut the DNA. These enzymes normally recognize something like 6 or so base pairs. How often would you expect these enzymes to cut a piece of DNA? Let's say a recognition site consists of 6 base pairs. At each position, you have 4 different possible bases. If we have a random sequence, and we assume that each of the bases occurs with equal likelihood, there's a 1 over 4 to the power of 6 chance, a 1 in 4096 chance, that any 6 base pair sequence will match that recognition sequence. You would expect the restriction enzyme that recognizes that site to cut on average every 4096 bases, giving you fragments that are on average about 4 kb. But you want to have larger fragments, more than 10 kb to clone into phage or Cosmids. So what you do is you do a partial digestion, meaning that you do not let the enzyme cut completely at every recognition site, and you can do this by using a small amount of dilute enzyme and stopping the digestion partway through. This way you end up with fragments larger than 10 kb, which you can then ligate into your vector. You would package the DNA into phage particles and then transform bacteria, then plate and identify the appropriate clones. When the phage itself is used as a vector for DNA inserted into its genome, the clones appear as plaques on a plate. When a Cosmid is used as a vector, the clones grow as bacterial colonies on plates containing a selective antibiotic. If, within that library, you want to identify and isolate a specific DNA fragment, say containing a gene that you want to study, how do you tell which clone carries that fragment? You might have hundreds or thousands of clones, each carrying a different fragment of DNA. Your textbook goes into an example of a method you can use to essentially search this library for a sequence. This technique uses DNA hybridization. If you are looking for a certain DNA sequence, you can chemically synthesize a short radioactively labeled oligonucleotide that is complementary to either strand of that DNA. If you have a plate that either has plaques from phage carrying cloned DNA fragments, or colonies of bacteria carrying Cosmids with cloned DNA fragments, you can transfer these onto a piece of nitrocellulose filter paper. DNA binds to the nitrocellulose wherever plaques or colonies had been transferred. You can block the rest of the nitrocellulose paper with non-specific DNA. DNA can be denatured into single strands by soaking the nitrocellulose paper in an alkaline solution. Then you can add your radioactively labeled oligonucleotide to the filter paper with denatured DNA. Because the nitrocellulose paper has been blocked with nonspecific DNA, the oligonucleotide will only bind where there is DNA from a clone that has a complementary sequence to which it can hybridize. You can wash the nitrocellulose paper and see where the radioactivity remains bound. This will tell you which plaque of phage or which colony of bacteria has the clone carrying your DNA of interest. That's it for this lecture. In the next lecture, we'll talk about cloning cDNA, we'll introduce the polymerase chain reaction, and we'll talk about methods for expressing cloned genes.